The reason we didn't is because honestly, we just didn't have a lot of time. I will remind you though a couple thoughts about chapter 5. These are my thoughts. 50% of it, of the first exam, is from chapter 5. So chapter 5 has a lot of representation on this first exam. So does chi-square too. In fact, if you know chapter 5 really well in chi-square, you could actually have a really good grade on the exam and you've only looked at one of 5 chapters. So really make sure you're good on this. But we had also spent some time defining the principle of what a monomer is and then quickly defining the principle of what a polymer is. And we had suggested that a monomer was when you had a single unit. We said a monomer was analogous to being just a subunit, one entity, one piece. A polymer, though, was a set of bounded or bound subunits. So a monomer is just one piece. The polymer is all those pieces combined together. My analogy that I was using the other day suggested that a monomer was like a letter, whereas a polymer was more like a word. And it was a letter that combines to form words. To go through an example, let's take the monomer A, so the A monomer. Let's take the T monomer and the C monomer, and we'll put them together. So there's the A, there's the C, and then there's the T monomer. So I have here 3 monomers. If I was to link them up, linking them up would yield a product that looks something about like this. I have formed in this process 2 new bonds that did not exist before. I'll highlight those bonds in yellow. So there's 1 new bond, and there's a second new bond. So 2 new bonds have been formed. So a new bond here and a new bond here. And if you've been keeping up with the class, you would know that this bond is more ionic or covalent in nature. Covalent because this is biology, and biology has mostly C, O, H, and N, with some sulfur and phosphorus to constitute something like 99% of all biology. So for me to suggest it's all covalently bound, that would be sensible. We also had established that this process would require the elimination or removal of 2 water molecules. This was collectively referred to as a dehydration reaction. And we had mentioned that through last week's notes pretty heavily, dehydration reactions. One in which you remove water to link pieces up. Your book used a synonym on a couple of occasions. That synonym was condensation. So a condensation reaction is analogous to a dehydration reaction. One in which you use the removal of water to link your pieces together. That's how that works out. So that would be the polymer making process. How about the polymer breaking process, though? It would be a direct opposite. Yes. Perfect. So let's take this entity. I'm going to draw out a polymer in a different way. There's 4, 5, 6, 7, 8, 9. So there are 9 monomers over here in this polymer. Yep. So there's 1 polymer that's right there. And if I was going to do a reaction in which I'm breaking down the polymer, the yield to this process, if I was able to break it entirely down, would be 9 individual pieces. There's 2, 3, 4, 5, 6, 7, there's 8, and then there's 9. So I have here 9 monomers. These 9 monomers are formed by the breakdown of a single 1 polymer. And Austin is correct. This would be definitely a hydrolysis reaction. We could be a little bit more specific in quantifying those. So how many water molecules would be needed to break up those 9 monomers, those 8 bonds? So 8. So I would need to feed this system a specific count, and that specific count would be 8 H2Os. And if I do that, then these 8 H2Os can break up the polymer into the 9 monomers, because I would be breaking the 8 bonds in it. The term hydrolysis literally means water splitting. So the term hydrolysis directly connects back to this idea or notion that you are taking a polymer and breaking it up into little pieces. And water is the required component, or a required component. So here's some thoughts about carbohydrates. First of all, they are used as an energy source. Okay, so they are pretty much like a quick energy source. You use these to facilitate your body's needs when you need quick energy. So like a commercial back in the day for Snickers bars was like an NFL football player would eat a Snickers bar before the big game, um, which would give them quick energy. Y'all recognize that commercial? What? Okay, so that's what they do these days? Okay, I haven't watched TV in a long time. But actually, when I used to watch TV pretty regularly, I watched a show called The Office. Y'all ever seen that show before? So it reminds me of this episode when they're supposed to do this big 5K or whatever. It's the Office 5K, and the boss, who's like, he's really just a fool, right? So this guy is like the office leader, and he's a little he's just a foolish guy. And so he's about to run this 5K, and he decides that the way that he needs to prepare for it is to carb load. Do y'all remember that episode? Do you remember what he ate? He ate like just this big mound of pasta, and he gets like I don't know hundreds yards, a couple hundred yards into it, and the next thing you know, there's a scene of him just vomiting off on the side. He obviously did not carb load correctly for his quick energy, um, but that's the idea of it. But there's more than that. Carbohydrates are a quick energy source, so definitely that's true. But I would say also they serve as markers for outside of the cell found in the membrane. And so, in memory of all the announcements we've been hearing today, I would say that a carbohydrate from a cell function on the outside is like an ID badge. They identify a body cell as being a certain type of cell. Each cell has a marker on the outside. A carbohydrate is that marker. Also, they serve in a structural capacity. They serve as structural components. I know that's true because we had an incident earlier in the class today where we had to kill a bug, and that's where the idea of the takeout, that there was that definition there that just could have been interpreted multiple ways. And so when the bug was squished, I suspected, was there a crunch? There was a crunch. Okay, you actually were crunching carbohydrates at the time. You were crunching a type of carbohydrate at the time, which was intense, but nevertheless. Next, the principle of a monomer. Carbohydrates exist at multiple levels. There's monomers and there's also polymers. The monomer for carbohydrates is essentially the monosaccharide. We mentioned that the other day. It is the entity, the most basic entity of the carbohydrate group. And we had alluded to that there was a group of monosaccharides we should be familiar with. That group contained glucose, it contained fructose, it also contained galactose. The connecting ingredient, if you will, between those three was that they all had the same formula, that being C6H12O6. So that formula is consistent between all three groups. I will tell you that easily glucose is the most preferred. I'm thinking like from human body perspective. Fructose is not so well preferred. It is, maybe you could argue, actually the least preferred, and in fact, it can be bad for you. And that's not just like you as in, oh, you're that one person. It's pretty much, it can be bad for just about anybody. When I think about these, I'm thinking about this picture. I'll go back to the notes, but here's a picture for you. This is a picture of high fructose corn syrup. This is what makes soda soda. It looks very honey-like and very syrupy. You've heard of like the fact it's like a syrup ingredient. You just have to bring that to the right temperature and mesh it with some water and some acid, and you have yourself soda, as in Coca-Cola, which that's probably not good for you. This is like what most soda is made out of. When I was doing my research for high fructose corn syrup earlier, I stumbled upon another picture of it. This was from a different guy. I'd like to show you his take on high fructose corn syrup. Well, he lets you know he seems to be very upset about high fructose corn syrup. That's his mind's way of designating the evilness that is high fructose corn syrup. That would be different. That would be a cane sugar would be soda, but that's made with just sucrose, which is part fructose, part glucose. That actually might be better for you than the refined high fructose corn syrup. Probably, and it would taste better. It's just more complex to make, and it's therefore more costly to buy. These three are all isomers of each other. They all have the same formula, but they just have different shapes, which means they have different properties. Like, for instance, your body likes to use glucose, but your body's not so good at using fructose. But there's more. I would be doing a disservice to the group of carbohydrates called the monosaccharides if I didn't include two other important monosaccharides, ribose and deoxyribose. The linking feature of those two, the reason I mentioned these at the same time, is because they both have 5 carbons, as opposed to the 6-carbon hexose sugars earlier. These have 5 carbons. Ribose is an important ingredient in making RNA. Deoxyribose is an important ingredient in making DNA. In fact, RNA stands for ribonucleic acid, ribo as in ribose. DNA stands for deoxyribonucleic acid, as in deoxyribose. Good so far? Yeah. That's where I was going to go to next. The ose ending that you see commonly at the end of these, ose, 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 and ose, really just connects back to the principle of carbohydrates. So anytime you see an ose ending, it's probably something to do with some sugar. Probably a monosaccharide, but could be a disaccharide too. Could even be a polysaccharide, carbohydrate-based. Yay. When I draw carbohydrates, oftentimes draw them in their naturally occurring shape. When we draw chemicals normally, we draw them in a line form because that's easier for us to do and to draw, but the common form of these monosaccharides is actually a ring shape. So I'm going to draw out the common ring structure of glucose because glucose is the preferred sugar, and I want you to see how I draw out those ring shapes. Draw out something like that. I could identify the number of carbons in this compound. We know it's 6 carbons because glucose has 6 carbons. There's carbon number 1, there's carbon 2, 3, 4, there's 5, and then lastly there's that 6 carbon right there. They're just not shown. In organic chemistry, you can draw things like this and actually get away with it because it's much simpler, given that there are so many carbons. So we've talked about monosaccharides, your single unit sugars. Ribose, deoxyribose, glucose, fructose, and galactose would all fit in that category. The next level, the disaccharide group. Disaccharides would be when you have multiple monosaccharides, actually 2 monosaccharides that are bound together. So it's when 2 monosaccharides are bound together. And understandably, you can only bond these 2 monosaccharides if you were to remove a water molecule. That would be dehydration that's responsible for making this happen. So if you take a glucose, you could bond that glucose to perhaps another glucose. When you do that, you would end up with a molecule called maltose. Glucose is C6H12O6. The other glucose, of course, is C6H12O6. So what would the formula for maltose, therefore, be? It's definitely C12. And then O11. And the reason it's not H24 is because you have to remove an H2O. It should have been 6 and 6. It should have been 12 O's, but it actually is 11 because you removed a water molecule. So maltose, a glucose and a glucose combined. Or you could take a glucose and you could bond it to a galactose. That's another monosaccharide. You would end up having lactose. That's milk sugar. Or you could take glucose. This is everyone's favorite. You can combine it with fructose. This makes sucrose. We know sucrose as being table sugar, as in I ate a lot of cookies this past weekend, so I ingested a lot of table sugar. And I would understandably form these by dehydrating between the two monosaccharides. So if I was to draw this in example format, here would be my picture of glucose. There's my glucose. Fructose is commonly shaped kind of more like a pentagon. So there's my fructose. They both have 6 carbons. And it's common for these molecules to have hydroxyl groups on them. So there's hydroxyl groups at multiple points, perhaps at that point as well. And so if I link them together, the only way I can link these entities is if I have to add or remove water to link them? Remove. So if I link them up, I would be removing a water molecule. That's through dehydration. And so I would end up with a molecule that looks something about like this. Polysaccharides would be when you have many monosaccharides that are bonded together. So you have multiple monosaccharides. Now, oftentimes, the monosaccharides involved in this bonding are glucose. So every polysaccharide has glucose that's just bound in multiple different ways. And those different ways of bonding glucose is what gives each polysaccharide its different properties. Your book goes through polysaccharides from the perspective of several examples. Actually, it's specifically 4 examples that you need to know. And you need to know pretty much enough about them, all 4 of them, to be successful in answering any questions about them. The first group that I'd like to identify with is the group called the starches. When we think of starch, we oftentimes think of these pieces of a vegetable that are worth maybe some large amount of energy. So a starch is a polysaccharide made of glucose, and this polysaccharide serves as energy storage for a plant. So this is how a plant stores its energy. A great example of something that's starchy would be like a potato. A potato is a good example of a starchy food source. But that potato is something that you yourself can use for energy. So I would have us know that humans can use this for energy. So we are energy users of starches, like a potato. That brings me back to the idea of that carb loading scene from that show. Yes. Glucose. In fact, they all have glucose in them. I've listed one of 4, and they pretty much all 4 of them are going to be glucose. It's just bound together in different ways. So starch is good. Here's another one. It's plant-based as well, but different properties. The next one is cellulose. Cellulose is another polysaccharide, but this one serves as a structural component in a plant. So this cellulose is what you would find like in the plant body, as they say. So when I think of a plant body, I'm thinking like literally like the wood structure. So the cellulose is it's the part of the plant that like It's like this part. It's like the wood part. You don't get much energy from cellulose. You get really almost no energy at all. A good example of something with cellulose would be like celery. Kind of got that plant crunch to it. Lettuce is the same way. Wood is the same way. In fact, they actually say, speaking of celery, they actually say celery is negative calories. You know why someone can say that celery is actually negative calories for a person? Right, like you spend more energy to ingest celery than you actually get from it. This cellulose product contains very little energy ultimately, but it's still important for you. You know that celery and any green vegetables are important for you because they provide you with the needed fiber that helps to regulate your digestive system, specifically the activities of your large intestine, so that you remove waste well. So both found in a plant, but different. Obviously, we can use both, but we really use the starch for our energy. Cellulose really just more for regulation of digestive function. One that you're probably very unfamiliar with is chitin. You're familiar with it, you just have never associated the word. Chitin refers to the outer covering in bugs and in mushrooms. When you squish a bug, it's got the crunch. That crunch is chitin. They have a skeleton that's made of this chitin. Their skeleton's on the outside of their body, as opposed to ours, which is an endoskeleton on the inside of our body. They have an exoskeleton. Their chitin, when you squish it, it's the crunch part. The last one, and probably for us the most important, but it's one you're not familiar with, is glycogen. What glycogen is pretty much the way that we store glucose molecules. It is our storage mechanism of glucose. It's like our glucose bank. It's when you have many glucoses that are bound together. I think of it like pretty much glucose temp storage so that I can use glucose later whenever I need it. So it is an animal fuel storage mechanism. That's what glycogen is. It's how we store fuels. We actually specifically store these fuels in our liver, in our muscles, and in our brain. That's interesting to consider. I mean, let's talk about muscles. What do muscles do for the human body? They contract, which causes the movement of the body or objects, right? Do you think body movement and movement of muscles is an energy intense process or like energy not intense process? Okay, so you need to have quick sources of energy, glycogen. It's like a bunch of glucoses connected together. You need in your muscles. How about your brain? Do you know how much the human brain weighs? Somewhere between 4 and 7 pounds, generally speaking. The human brain, it's like a fraction. It's like 3% of a person's actual weight in general, maybe 4%. No more than 5, probably. This brain uses, though, 20% of your daily energy. You need a lot of glycogen in your brain because you use a lot of glucose there. It's also in your liver, which is the hub of pretty much where you store energy in your body. All 3 of those sites is where you would keep glycogen, and you keep glycogen there because you rebuild it each day. You need to know that glycogen is an entity, a temp storage, that's actually rebuilt each night. So if you decide to pull an all-nighter studying for the exam, probably you would be doing yourself a disservice because you would not be allowing your glycogen stores to rebuild, which means you would not have the energy in your brain that you need to function correctly in the test. That's why good night's sleep is a requirement. Without it, you don't have your storage units for glucose. So that's all that I have for polysaccharides. We'll move on to lipids, but we will not be able to get through our lipid discussion today. We'll be able to get started, and that's about it. When I think of lipids, I generally think of the idea of having entities that are fat-containing. So we oftentimes associate the word lipid with being pretty much like fats. And when we think of the word fat, oftentimes we suggest that that means that we're about to have a discussion about something that's very bad. We associate fats as being bad. But I would argue that they're bad if they're excessive and excessive in the wrong type. For biological functioning, I think it's important that we land on this idea that they are definitely required, and not just that, but they are good more times than not. They become bad when they are too much of the wrong type. That's where it becomes typically pretty bad. There are multiple types of lipids that I think we have to be familiar with. This is how we'll approach teaching lipids. We'll get through the first group today. One group is called the fats. More specifically, you might have heard the term triglycerides before, like the common fats. A second group is the group called the steroids. We oftentimes suggest that steroids are like when we think testosterone, we think like muscular and like the illegal type of steroids for like sporting events. But estrogen is a steroid too. Or you could also include cholesterol. That's a steroid by definition as well. You could be injected in chickens. There's steroids that are just, I don't want us to think steroids are bad, either they make things grow or that's all about muscles. Steroids are, there's a lot of different things that are classified as steroids. And our last group is the phospholipids. All of these three are three types of the lipid group. We'll talk about the first group, which are your fats today. But before that, we have to identify some general properties. General properties of all lipids. What are some things that we can say that all lipids have that other entities, other groups like proteins and nucleic acids and carbohydrates don't? One thing is that all lipids have long carbon chains. They're composed of generally long carbon chains with very little more than just hydrogen attached to it. Oftentimes, that allows these entities to have lots of energy. These are high energy compounds, generally speaking, therefore more of the long-term storage. So when we think of carbs, we should think of quick energy, but when we think of fats, we should think of a long-term permanent storage energy. Another important concept about acids is that they have no affinity. For water. They're therefore viewed as being hydrophobic. That's the term we use to describe the relationship between those types of compounds. These hydrophobic compounds have pretty much no mixing with water based upon the nature of those compounds. I do want to get through that first group today, which is your group about triglycerides. If you go get your blood tested to see all your cellular counts, your doctor may call them triglycerides. The more proper name is the term triacylglycerols. But the two terms are ultimately kind of synonymous. Regardless of what word you choose to use, I will tell you that every triglyceride is composed of a 3-carbon head. That's referred to as the glycerol component. And then also 3 fatty acid tails, which are the long carbon chain component. So 3 fatty acid tails, I'll designate in red. And then let's say that the 3-carbon head, I'll designate here in green. So the way that it works is you have your glycerol head. There it is, it's 3 carbons. Then you have your 3 fatty acid tails, which I need to draw out in red. I'll draw out the first one being somewhat specific. There'd be a bunch of H's here. It's a lot of H's I have to draw, and I won't draw them all in because you can kind of get the idea. And I would need to draw something like 20 carbons. That becomes quite tedious to do. So I'll just designate that I would need 20 carbons, and I'm going to leave it at that, and then I'll show you the shorthand way. Here's the shorthand way. Drawing that skeleton model is an easy way to talk about and get all your carbons included without it taking forever amounts of time to draw. The theme, though, here is that it's this head unit with a lot of carbons attached to it. And carbons, when they're bound together, are worth a ton of energy, so it so works out that this entity has a ridiculous amount of energy for its overall size. We have here shown those glycerol heads, and then lastly on the right-hand side, the fatty acid tails.
How does that squiggly line?
Each dot right there is a carbon. So you just get 20 squigglies, and you've kind of got it made. Now there can be anywhere between actually 16 and 24 carbons depending on the type of fat. So this number right here does vary, but we won't get into that in this class. I will say that the way that you take a glycerol head and then your fatty acid tails and put them together is through dehydration, which is sensible. We've seen that before. Dehydration reactions, we know, is what can connect the glycerol head as well as the tails. This bond that's formed is called an ester linkage. And I do want you to know that that ester linkage is something that does exist. For tomorrow, I'll tell you that it works out that these triglycerides may be either saturated or they may be unsaturated. And we'll see that both could be good, both could maybe be bad. It kind of depends on the amount, the type, and then the subtype of each of these. But we'll talk about that tomorrow. This would be a good spot to leave off at.